Hello, everybody. I am Brooklyn Jones, and today we're going to present a couple learning objectives from Chapter 5. So I'll be talking about this first one, phago, endo, and exocytosis. First one is phagocytosis. So first we're going to start out with a cell. This is a bacterium, and these are lysosomes. This is the nucleus. Essentially, we've got the cytoskeleton which extends, and the edge of the cell membrane will completely envelop the bacterium, which binds onto the surface membrane. It forms a vesicle inside of the cell. The lysosomes will bind onto that vesicle, and it digests the bacterium. So that's phagocytosis. This is receptor-mediated endocytosis. Essentially, it's a little bit of a different process. We've got some ligands here in the ECF. Then we've got the receptors right here. The ligands will migrate towards the clathrin-coated pit, which is right here. These pink lines are the clathrin, and sometimes instead of clathrin, cells will use caveolae instead, which are caused by lipid rafts. The clathrin will help form these vesicles here, and the cell is now going to be in the vesicle will now be in the cytoplasm. This vesicle will split into two parts. The part with the receptors, or with the ligands, are going to go into their next destination, so the Golgi, for example. And then the other half, with the receptors, will migrate towards the surface of the cell. The RABs and the snares are proteins. The RABs will help dock, and the snares will help the vesicle fuse into the membrane, and the process starts all over again, just like how I depicted earlier here. So at this point of the process is when exocytosis starts, and the first part was endocytosis. So here's a little recap of what we just talked about. Phagocytosis engulfs the bacterium. Endocytosis and exocytosis are opposites. Endocytosis is about clathrin, how it invaginates, it pinches off, and the vesicle splits into two different sections. And the exocytosis is when it fuses back into the membrane and releases the contents into the ICF.
Hi, my name is Elliot Chambers, and I will be talking about the next learning objective, which compares the three types of transport of different molecules across the epithelial cells. So the three types of cell transport or molecule transport is paracellular and also transcellular transport. We'll talk about transcytosis in a moment. So paracellular, when you break down the term, it means besides the cells. So these little molecules will go through these tight junctions, and they will go from the lumen, such as the intestine, and go to the extracellular fluid. And this one's not as common, but it does happen once in a while. Transcellular transport is where these molecules, they go through the apical side of the membrane and go through the basolateral side. And this can happen the other way. Going this way is absorption, and going this way is secretion. Now, the next mode of transportation of molecules is transcytosis. This process utilizes concepts that you have previously learned with endocytosis and exocytosis combined. In this case, these molecules are proteins because they're much bigger and they're harder to transport by themselves, so they need some way for them to be transported through the membrane, and that's done through vesicle formation. Here you can see that we're in the plasma, so you can see these red blood cells, and these are the plasma proteins. So when proteins need to be transported across, they first go through a process called endocytosis, where they create the vesicles. And then once they go to the other side, using the microtubules that make up the cell's cytoplasm, they're able to fuse onto the other side, and they're able to expel the proteins into the interstitial fluid.
Hey guys, my name is Israel. I'll be teaching the last two learning objectives, specifically about the resting membrane potential and the Nernst equation. So to begin with the resting membrane potential, the definition is the electrical difference between the ICF and the ECF. So here's an example of an experiment done to help us figure out what the resting membrane potential of a cell is. So the cell here, one The cell here is hooked up to an electrode, which is connected to a voltmeter, as well as the ground, or what the outside the ECF is. And the voltmeter is able to conduct millivolts for us. The inside of the cell usually is negative, so it makes sense that the inside of it is -70, and in the outside, because it's ground, it's 0. So through conducting this experiment, it helped us understand that the resting membrane potential, the resting membrane potential of a cell is usually -70 millivolts compared to the outside. Then, understanding this, we can apply it to this question. The question is, how do I find the equilibrium potential of an ion? The equation that we use to find the equilibrium potential of a specific ion, because in the experiment, it was for all the ions included in a cell and outside of a cell, we consider using the Nernst equation. The Nernst equation is specifically 61, which refers over to the 37 degrees Celsius, using a bunch of constant factors. We're able to figure out that 61 over Z, which is the charge, which is positive or negative, whichever one with the ion, log over ions out of the cell and into the cell. Then we're applying it for an example with potassium. So usually potassium, the millimoles inside of the cell are 150 and outside of the cell are 5 millimoles. We plug all the factors into the equation and we get around -90 millivolts. So the electrical potential of the potassium ion for the cell is -90 millivolts, which makes sense as we compare it to other graphs. So that's how you apply the resting membrane potential and the Nernst equation to figure out the electrical potential of a specific ion. And that's it. Yeah.